Let us start with topic contacts between adjoining cells. Now here you can see an unspecialized contact between two cells. Here this is cell membrane of one of the cell, and this is another cell membrane of another cell. And the gap between them is about 20 nanometer, and these are connected with the help of CAMs or cell adjoining molecules. And these CAMs are attached to transmembrane or integral protein, what we saw in other class, then or intermediate protein. And to this intermediate protein, the cytoskeleton fibers are attached. So we can say the transmembrane or integral protein is joining or linking the CAM protein with the cytoskeleton fibers. Now let us see types of CAM or cell adjoining molecules. There are of two types, calcium dependent as well as calcium independent. Calcium dependent are like they depend on calcium for adhesion of two molecules. There are of three types. Cadherins, they are seen in epithelium cells. Then selectins, they are seen in marginal cells, example like leukocytes. Then integrins, they are seen between the cells and intercellular substances, and there are about 20 types. Now calcium independent, neural cell adhesion molecule or NACM, NCAM, they are seen in nerve cell, and intercellular cell adhesion molecules, ICAM, seen in leukocytes. Now let us see about types of junctions. Types of junction, the first junction is adhesive or anchoring junctions, and there are of three types: adhesive spots, adhesive belts, adhesive strips. Then the modified adhesive or anchoring junctions are which they are the connections which connect the extracellular material, like examples are hemidesmosomes or focal spots. Now second type of junction is occluding junction, which is also known as tight junction or zona occludens. Then communicating junction or also known as gap junctions. Now let us start with the first type, which is adhesive or anchoring junction. Now adhesive spots in adhesive anchoring junctions, the first type is adhesive spots. These are known as desmosomes or maculae adherens, and here you can see the picture. The gap between two cell membranes is about 25 nanometer, and we have spikes like arranging like comb, which are desmoglin and desmocoglin, and the actin fibers are present as cytoskeleton fibers are actin filaments here, and the transmembrane or integral protein is desmoplanktins. The CAM is made up of desmoglin and desmodecolin, which are a part of integrins, which are calcium dependent. Now these are seen in epidermis, skin, and in cardiac tissue or intercalated discs in the cardiac tissue. Now second type is adhesive belts, also known as jonula adherens, and it is a form of continuous band passing all around the apical parts of epithelial tissue. And here cams are present. A type of cams like cadherins are present, which are also calcium dependent. Now adhesive strips, fascica adherens. These are similar to adhesive belts, but they don't form a continuous band around apical part, but they form short strips like substances to attach the molecules. They are seen in smooth muscles, intercalated discs of heart, and junction between the neuroglial cells and the nerves. And now hemidesmosomes. They are basement epithelium cells. They are seen in basement epithelium cells, which connect the basement epithelium to connective tissue. Then transprotein are keratin filaments, and CAM are integrins. Integrins is also calcium dependent, and filaments which are present are keratin filaments. Now focal spots. Focal adhesion plaques or focal contacts, they are known as focal spots. They help in the attachment of local adhesion of cell to extracellular matrix. They are seen in leukocytes or a vessel wall, where CAM is integrins, a calcium dependent, and intermediate protein is it may be alpha actinin, vinculin, or tallin. Now the second type is also known as occluding junction or tight junction or zona occludens. They're seen in they're similar to adhering junctions. They're also attached in apical parts of cell. So they're also in apical part of cell where cells are in actual contact with each other. They act as diffusion barrier. The CAM's presence are claudins, which are calcium dependent, and actin fibers are present as cytoskeleton fibers. Then they are seen in epithelium, endothelium, GIT, respiratory tract, and blood-brain barrier. Sometimes the junction is incomplete, and it is also called it is called as leaky junctions, and it is seen in PCT where the junction allows some certain molecules or ions like potassium, calcium, chlorine, sodium, etc. Now let us know about junction complex. Near the apical part of epithelium cells, there are three junctions present at a time, like zonula occludens, zonula adherens, and macula adherens. That is, they are tight junction and hemi adhesive belts and adhesive spots. These three, the tight junction, adhesive belts, adhesive spots, are together present at the apical portion of epithelium cells, forming a junction complex. So this is known as junction complex. Sometimes zonal occludens are replaced by leaky tight junction. In case of PCT or proximal convoluted tubule, we can see this. Now third type or last type of junctions is communicating junction or gap junction. It is also known as macula communicantis. Cells are actually not in contact, they lie closely to each other, where the gap decreases from 20 nanometer to 3 nanometer. So they are close to each other, but they'll not touch each other. Then change in pH or calcium ion concentration can close the channels of gap junction. It is seen in cardiac tissue like intercalated discs, GIT, and neurons. Now here you can see the cam of communicating or gap junction is made up of connexon, where each connexon is made up of 6 connexins. So we have 12 connexins to make one gap junction. Now let us move to cell organelles. Now cytoplasm is of two types. We know cytoplasm is a fluid matrix-like substance which is present inside the cell. So it is of two types. If it is inside the channel or membrane-bounded organelle, it is known as vacuoloplasm, or it is outside the channel, it is known as hypoplasm or cytosol. Now endoplasmic reticulum. It is the first organelle we are discussing here. Now endoplasmic reticulum is of two types. You already know that: rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum are associated with ribosomes, so they have rough surface. So the name comes from it. So it helps in it has main function as protein synthesis. Now smooth endoplasmic reticulum will help in further processing of protein which is synthesized in rough endoplasmic reticulum, and it also helps in synthesis of lipid and especially the membrane phospholipids. Now products which are synthesized by endoplasmic reticulum are stored in channels within the reticulum, and ribosomes and enzymes are on outer surface of membrane of the reticulum. Now second organelle is ribosomes. They are present individually. As we saw already, the ribosomes are already present in rough endoplasmic reticulum. Besides that, they are also present in the cells individually or grouply. So if they are present individually, they are known as monosomes, or in group they are called as polyribosomes or polysomes. Now each ribosome has protein as well as RNA, and it is about 15 nanometer in diameter. Now ribosome is made up of one large and one small subunit which play important role in protein synthesis. Now third organelle is mitochondria. Here you can see the meaning of mitochondria. Mitos granule or chondrin is rod. So it according to its meaning, it appears as a rod or granule when we see in the electron microscope. It has size about 0.5 to 2 mu meter, and there are more There are more number of mitochondria in cells if the oxidative metabolism is high in the cell. And all mitochondria are formed in the fertilized ovum, so they're they're entirely from the maternal origin. Now here you can see the parts of mitochondria. First we have outer smooth outer membrane, and the middle and the second is inner membrane. In between these two membranes we have intermembranous space, and now this spokes like thing is known as cisternae. Then matrix is present, which comprises some enzymes, RNA, DNA, etc. Now let us move to the fourth organelle, Golgi complex. Golgi complex is similar to endoplasmic reticulum, and the material from the endoplasmic reticulum are sent to Golgi complex with help of vessels. Golgi complex is near to the nucleus and extends till the cell membrane or cell membrane or plasma membrane, and it is divided into three phases: cis phase, medial phase, and trans phase. And the pathway of material passing from ER to the Golgi complex is as follows. Here, first the rough endoplasmic reticulum, then the material moves to smooth endoplasmic reticulum, then it enters the first phase, which is near to the nucleus, cis phase, where the proteins are phosphorylated. Then the medial Golgi complex. Here the sugars residues which are left already are added to the protein, forming protein carbohydrate complex. Now finally the transphase. In transphase, the sugar residues are also added in transphase as well, and the functions when it reaches transphase are proteolysis, that is they convert the inactive proteins to active proteins. Then various substances are sorted and packed in the vesicles. Then vesicles may be secretory vesicles, lysosomes, or vessels which are meant for transport, or excreting vessels, etc. Then the fifth organelle is membrane bounded vesicles. Now there are many types of membrane bounded vesicles. The first one is phagosomes. They are formed from ER or endoplasmic reticulum, which help in engulfing of foreign materials including bacteria, virus, and all. And this process is all known as phagocyte. We already studied in the back previous lecture. Then pinocytic vessels. It takes fluid inside the cell and some soluble materials, which is also known as pinocytosis. Then exocytic vessels, it is called as reverse pinocytic. It removes or exports the material from cell to outside the cell. It is called the process is known as exocytosis. Now secretory granule, it is derived from Golgi complex and appear different each time due to type of substance present in it. It helps in secretion. Now, other storage vessels like lipids, carbohydrate may be present, which are bounded and present inside the cell. Now, this lysosome. Lysosome is which help in destruction of unwanted material, or have enzymes like proteases, lipases, carbohydrates, and acid phosphatase. And it follows acid vesicle system. The membrane of these lysosomes have H plus ATPase, where the membrane act as uh, H plus acidic pump, where it can reach up to pH of 5, which creates acidic medium inside the cell. Now let us know about the stages of lysosome formation. This acidic hydrolase enzyme from ER reaches to the Golgi complex where the vessels are packed, but they are inactive due to lack of active acidic medium. So the first primary lysosome or Golgi hydrolase vesicle is here. And the secondary lysosome is formed where the vesicles fuse with endosomes. These endosomes are from cell membrane which produce acidic medium. So this fusion is called as endolysosome, which is also known as secondary lysosome. Then the final lysosome is formed while the H plus ions are pumped into vessel. Already said the membrane acts as H plus pump, so the cell membrane will pump H plus inside the cell and to create the acidic medium. Now this acidic hydrolase enzyme is converting from inactive state to active because it has acidic medium. So this activate and mature the lysosome. Now lysosome is formed. Now lysosome can be fused with phagosomes to destroy all the phagos, and this is known as phagolysosomes. And if they fuse with pinocytic cells or vessels, they're called as multivesicular bodies. After digestion of waste material, they are sent out by exocytosis, but some may be present in cells as residual bodies. Now, lysosomes are present in every cell except the mature erythrocytes, and they are prominent feature of neutrophil leukocytes. Now, lysosomal glycogen disease, where the glycogen is absent, and Tay-Sachs disease, where the lipids are absent. These occur due to absence of specific acids due to genes. Now, peroxisomes, they are similar to lysosomes. They produce hydrogen peroxide to detoxify the cell's contents, and they are majorly seen in liver and renal tubules. Defect in enzymes of peroxisomes can lead to metabolic disorder and abnormal lipids in some cells like brain and adrenal cells. Now let us move on to the sixth organelle, cytoskeleton. It has many types. The first is microfilaments. They are 5 nanometer in diameter. They are made up of actin, and in actin also we have type like G actin, where several G actin join together to form F actin or the actin filaments or the microfilaments. Now actin create a meshwork called as cell cortex, and this meshwork are held together due to filament protein. These actin filaments can be rearranged, and this is what makes cells to alter its shape time by time. Now microtubules, they are 25 nanometer in diameter, and the basic constitute to microtubules is protein tubulin. Two types like alpha and beta tubulins. The chain of tubulin form protofilaments and they are stabilized by microtubule associated proteins or MAPs. The microtubules are seen in centrioles which have microtubule organization center. Now let us see the functions of microtubules. They provide stability and prevent endoplasmic reticulum collapse. They facilitate transport with its cells and help in with the help of digonin and kinesin protein seen in axons, which keep the cell cylindrical in shape. And then in cell division, microtubules from mitotic spindle. Cilia are made up of microtubules which help in locomotion. Then intermediate filaments. With respect to the size, intermediate filaments are between the microfilaments as well as microtubules, because the size of microfilaments is 5 nanometer, whereas microtubule is 25, but the intermediate is between them, that is 10 nanometer. Now proteins vary from cell to cell in intermediate filaments, like cytokeratin is present in epithelial cells, neurofilament is present in neurons, desmin protein is present in muscles, and glial fibrillary acidic protein is present in astrocytes, laminin is present in lamina, and vimentin is present in many cells. And now the functions of intermediate filaments. They link cells together both extracellularly as well as intracellularly. They modify to form keratin in skin and form major constitute of the hair and skin. Neurofibrils help in maintaining cylindrical shape of axon, and then nuclear lamina has intermediate filaments. Now centrioles. It is series of microtubules arranged in circle. A group of which have 9 groups and each have 3 fibrils. These help in mitotic spindle formation, and cilia, flagella, and some special progressions are also formed with the help of centrioles. So we can see all have 9 plus 2 arrangement. This is because centrioles also have the 9 plus 2 arrangement. Thank you.